Good morning, everyone. This is a very unusual way to speak in a conference. So I would like to start by thanking the organizers for inviting me. I have to say that at the beginning, I thought it was going to be a 15-minute talk, but then I realized that it is a 45-minute talk. And it is for me a great privilege to go over the research we did in the lab in the past 10 years, but I will insist more on studies that are in press and studies that are in process of being published. So I hope you are all well. These are special times. I know it's not easy for everyone, so I really hope everybody is fine at this moment. These are my potential conflicts of interest. I will not go through, but they are listed here in the sake of time. These are the people who are working in my lab. I will not go through the list, but I would like to say that I would not be able to present anything today without their great work. I would like to thank them and to wish them to have a great career. Some of them already left the lab, so and are involved in other challenges. So I really thank them for their contribution and their wonderful friendship that they establish in the lab. I'm from Romania originally, so I know this delta of Danube a bit. This is a great reservoir of water and also unique with flora and fauna. What is interesting is that this river crosses so many countries in Europe. It's a huge river, everybody knows about it. And what it's less known is that this is the small well, the source of the Danube in the Black Forest in Germany. So I would like to make an analogy because a lot of times in HIV research in the HIV reservoir field, we try to look for a major HIV reservoir, and sometimes something small can give a rise to something big. So because of certain properties. So we in the lab work a lot with Th17 cells, and I will try to explain you why. From the beginning, I would like to tell you that they have some unique features. They are located at the portal sites of HIV entry. They are highly permissive to infection in vitro and in vivo. A fraction of them is long-lived and can harbor HIV reservoirs. And we know from studies in the field of autoimmunity that the pathogenic Th17 cells express drug efflux pumps that makes them refractory to multiple anti-inflammatory molecules. And we can make an analogy to antiretroviral drugs. This is something we don't know, whether or not the penetration of antiretroviral drugs is optimal in these cells. So as I said, I'm from Romania, and I would like to give a hint about my curriculum just for students that may be wondering how we manage through life. So I got my inspiration in immunology from the University of Bucharest. I was very surprised to know that cells are located in so special structure in lymph nodes, and I was really very keen to understand more about this. Then I had the opportunity to go to Paris and study more, to have a master degree and then a PhD in starting to work in the HIV field at the moment where 3 therapies started to prove efficacy in the treatment of people living with HIV. Then I moved to Boston, where I wanted to study more neuropathogenesis, and I was working with Anna Gabuzda, and I should say that I had another privilege to work in the department of Joe Sodrowski, were full of virologists that were thinking at viral replication and in deep mechanistic ways. And at that time, multiple discoveries were made, including TRIM5 alpha. And I was an immunologist lost among virologists, but I think this this helped me in orienting my my research project, my research career in the future. So we published a few works. I was interested in monocytes, and what I was impressed with the conference I participated about the zip codes of cell trafficking. So I understood that tissue-specific trafficking is essential for understanding immunology and later virology. So then I met Rafik Sekali in a moment where I was searching to transition from postdoctoral fellow to a principal investigator. It was a dream at the time. I didn't know I was able to do this. So in 2004, he invited me in Montreal, and I was presenting him some work where we found that chemokines produced by CD16 positive monocyte-derived macrophages will activate T cells, and specific T cells that express specific chemokine receptors, and through this activation, cells will be more permissive to infection. So it was a kind of immunological synapse. And then Rafik one day in 2005 sent me an email. You should come work in Montreal, and I think this is the concept you should bear in mind, working on chemokines as immunotransmitters. So I was passionate already about cell trafficking and understanding how tissue-specific immunity is important for a disease outcome. So I was happy to get a fellowship and start my lab, and I would like to thank Cécile Tremblay, who I think is presenting just before me at the virtual meeting, to say how much I appreciate that at the very beginning she decided to share a bench, and my lab was working on that bench, and we in fact for 3 years worked on that bench and generated some results so that we could renew my first CAHR application. So this was a fantastic adventure, and so Cécile is a major contributor to my success, and we continue to collaborate. So I show here as a figure from a review article written by Françoise Barré-Sinoussi, Nobel Prize for the discovery of HIV, where one can see how many discoveries were made that allowed the scientific community to understand in detail the viral replication cycle, the restriction factors that act at different levels, and especially the drugs that could be designed to block different steps of this replication cycle. And we know all that antiretroviral therapy changed the face of HIV/AIDS into a chronic, manageable disease. We know there is a lot of information. This is a slide from a paper published by Brass et al. In Science in 2008 that revealed through scRNA functional genomic screen so many host proteins required for efficient HIV replication. So in the context of this ocean of information, one can wonder, what can I add more to this? So one thing is very sure is that antiretroviral therapy is not perfect. We know this as soon as people stop therapy, there is what is called a viral rebound. So, and we understood in the recent years more and more what's the reason for this. At the beginning of the antiretroviral therapy, we thought in a few years we will reach the cure. This is not true. We know that the long-lived reservoir cells can harbor these reservoirs. We also know from some studies, not totally accepted by everybody, that maybe in some tissues antiretroviral drug penetration is not efficient, and maybe also in some cells is not efficient enough. And then a fact that is really clear is that antiretroviral therapy doesn't act on HIV transcription. So this allows residual viral replication in ART-treated people living with HIV in certain compartments. So I'm far from being a specialist in HIV transcription, but I know from textbooks and from most recent reviews, and here I'm citing a review of one of our collaborators, Brendan Bell, that viral replication is complex, viral transcription is complex. There are huge machineries involved. viral proteins such as STAT are key for making this machinery work, but also multiple host cell transcription factors. And I hope I'm going to be able to convince you at the end that in addition to host transcription factors such as NF-kappa B and NFAT, the transcription factors of the circadian clock machinery are also involved in this process and can be targeted therapeutically. So Th17 cells, it's a big story of discovery. It's a long venue. It started in 1986 with the initial Th1, Th2 hypothesis by Moshman and Kaufman. And then with the years, with the experimental approaches, especially in fundamental immunology, especially in mice, we understood that something else may exist. And people finished by cloning IL-17, by cloning IL-23, by finding the raw gamma T or raw C2 in humans as the main transcription factor for Th17 cells. And what also is interesting is that we were able to identify surface markers for Th17 cells. And I'm mentioning here studies by the group of Napolitani and with Frederica Salusto that started in 2007. And for RORC expression, I would like to highlight the role of the group of Dan Littman that was a pioneer in the Th17 field. So now this is a review from 2016, but I think this is just to reflect the complexity of the T cell heterogeneity. We know now there is a lot of plasticity and flexibility in the differentiation program, especially for some cells such as Th17 cells. So why these cells are important is that they are kind of adaptive immune cells that make the transition between innate and adaptive immunity. They are located in mucosal sites and they contribute, among other cells, they are not the only ones, to the homeostasis of the intestinal and respiratory and skin barriers. So if we talk about intestinal or respiratory barriers, we have here epithelial cells that when there is homeostasis, pathogens that are present in the cavity cannot penetrate into the systemic circulation. But if there is an alteration of this barrier, then we can have the process of microbial translocation. And this concept of microbial translocation was highlighted strongly in 2006 by Brenchley and Dweck. This was something that was known by clinicians, also other scientists, that HIV infection was associated with intestinal and also a pulmonary, disease. But in 2006, we understood better and we got experimental demonstration of the concept of microbial translocation in patients. And it's very well established nowadays that Th17 cells are depleted from these sites, from gut-associated mucosal tissues, and they remain depleted even in patients that are detected very early and treated very early. So now there are different mechanisms that were proposed during the years to explain the depletion of Th17 cells in HIV, but also SIV infection models. And I will not go through all of this. I would like to insist on the mechanisms that contribute to their permissiveness to infection in this presentation. So as I said, a major contribution in the field was the identification of surface markers for T cells that are polarized in different ways. So I think this is a wonderful paper that was published in 2007 by Acosta Rodriguez et al., where they were able to identify cells specific to CMV, to extracellular parasites, to Mycobacterium tuberculosis, and Candida albicans, based on their expression of CXCR3, CCR4, and CXCR3 in the presence of CCR6, or CCR6 in the presence of CCR4. So they were also able to tell that these cells are polarized in Th1, Th2, Th1, Th17, and Th17 cells. For us, this was an important progress because we wanted to study the diversity and the differential permissiveness of CD4 T cell subsets to HIV infection. And since these Th17 cells are located in the gut and they are depleted, it was easy for us to hypothesize that these cells are highly permissive to infection. So this is what I started to do in 2006 when I started my lab, and to look at the different permissiveness of cells, T cells to infection. And we were able to demonstrate that Th17 and Th1, Th17 cells were highly permissive to both R5 and X4 viruses, while Th1 cells will be relatively resistant, with Th2 cells being permissive to X4, but not R5 viruses due to the absence of CCR5. So this was our first contribution to the field, and my switch from myeloid cells to T cell biology. Of course, we got a lot of criticism, and a lot of people were still not convinced about the use of CCR6 as a marker of Th17 cells, and multiple groups, including the one I'm mentioning here by Unumat and Mark Sundrud, tried to clarify this problem, and they could demonstrate that in fact, CCR6 is a marker for Th17 cells able to acquire Th17 functions, meaning that they could sort cells that were CCR6 positive and were not expressing IL-17 after a short activation in vitro. And these cells, by culturing these cells, they were able to see that these cells started to produce IL-17. So indeed, then studies by Dan Littman and others demonstrated that the differentiation of cells into Th17 cells goes through two steps. One is specification of polarization with transcriptional reprogramming, the acquisition of certain surface markers. And then it's the second step, which is the induction of effector functions, including IL-17. So we went further into studying the heterogeneity of Th17 subsets, and by associating CCR6 with CCR4 and CCR3 expression, we observed that in addition to the subsets described by the group of Napolitani with Federica Salusto, Th17 and Th1, Th17, there were other subsets of CCR6 positive cells that were not studied, and especially in the context of HIV infection. And these cells, double negative, CCR6 positive, are the most predominant in the CCR6 positive pool. So we did a lot of cell sorting and culturing and transcriptional profiling in vitro, and we worked with cells from infected patients on ART and did a viral outgrowth assay, and we were able to show that these double negative CCR6 positive cells are unique in their production of cytokines. They have lineage flexibility as many Th17 cells. They are not depleted in people living with HIV treated with ART. We were able to show in collaboration with Steve Dix and Nicolas Chamon that gave us access to lymph nodes, that these cells were enriched in the lymph nodes of the patients, and they were permissive to HIV infection in vitro. And also we could outgrow virus from these cells in all the donors we tested, although for the other subsets, it was a donor-dependent phenomenon. So this is it about our focus on Th17 heterogeneity. We decided then to keep it more simple and to focus on CCR6 as a marker of Th17 polarized cells. Because these cells are able to go into the gut, we knew that they were permissive to infection in vitro. And I think what stimulated a lot our interest in going further into studying what molecular mechanisms make these cells permissive to infection and how can we design therapies that are specific to these cells was a study published by the group of Tom Hope that demonstrated that Th17 cells are the first targets of infection after a vaginal challenge in a SIV infection model. So then we decided to see if this is the case also in ART-treated individuals. But I will go, so I will show you how we we went into this question. So at that time, we didn't really think we were able to do this, because sorting the small subsets of cells from the gut, it was not an easy task. But we had a collaboration with Barbara Shacklett, and of course the wonderful collaboration with Jean-Pierre Routy and the nurse José Girard, and all the team of Jean-Pierre Routy helped us to get access to sigmoid colon biopsies and match the samples from blood, and we were able to isolate memory CCR6 positive and CCR6 negative from PBMCs and colon in a cohort of ART-treated people living with HAV. So here I'm showing the fact that the CCR6 positive T cells are recruited into the colon of patients, and their frequency is higher than that we observe in the blood. So there is an enrichment. And then we were able to measure GAG HAV DNA in cells we sorted, CCR6 positive and negative, and we could see that there is an increased level of HAV DNA in the CCR6 positive versus negative T cells in the blood and in the colon of these patients. So then we went further to study molecular mechanism of HAV permissiveness in these cells. Something that was very attractive to us was the idea of retinoic acid. Retinoic acid is an elixir for gut homing. It is produced by dendritic cells in the gut, which will degrade vitamin A, metabolized vitamin A, into retinoic acid. And it is known from fundamental immunology that retinoic acid will imprint cells with gut homing potential by regulating the expression of markers such as CCR9 and integrin alpha-4 beta-7, which are able to help cells localize into the gut. We also knew that this retinoic acid is a nuclear receptor, is binding to a nuclear receptor, the retinoic acid receptor alpha with RXR together, that will bind on retinoic acid responsive elements of multiple genes. And what is very important to notice is that the HAV promoter includes retinoic acid responsive elements. So this was the justification for our interest in studying retinoic acid. And we wanted to know whether CCR6 positive T cells were still more permissive to infection than CCR6 negative, even in the presence of retinoic acid. So in this figure, I'm just showing one donor where we looked at different levels of different transcripts, HAV DNA transcripts, early, R5, complete, GAG, and integrated HAV DNA. In cells from infected with a transmitted founder or with a lab-adapted NL4.3 BAL virus. And as you can see in both cases, we can see an increased reverse transcription and integration of the virus in CCR6 positive T cells exposed to ATRA. So I will not present all this paper, it's published in GCI Insight. What we found was that mTOR was one molecule overexpressed in CCR6 positive T cells after exposure to ATRA. So we decided to study the role of this molecule in HAV permissiveness. While we were finishing this work, a paper was published at the end of 2016 showing that mTOR controls HAV transcription. So since this was the case, we decided to add a new evidence to our work that was already quite complete, showing that mTOR inhibition using the ENK128 will reduce viral outgrowth from CCR6 positive T cells of ART-treated people living with HIV. And what was interesting to observe was that it seems there is a difference between people that are treated long-term with ART versus those recently infected, where we can also harvest outgrowth virus from CCR6 negative. But in people that were treated very long-term, it seems the reservoir is mainly segregated in CCR6 positive T cells. So we are now trying to do a study cohort to include a significant number of donors to allow conclusions. So it was interesting also to determine whether indeed T cells that infiltrate the gut of arthritic people have a retinoic acid exposure signature, and this was true. And we could see increased levels of CCR5, integrin beta 7, and phosphorylated mTOR in CCR6 positive T cells from the colon and not from the blood. So this was an interesting observation, and we concluded that CCR6 positive T cells are transcriptionally programmed to become HAV targets, and that by blocking mTOR, we may limit HAV replication and transcription in these cells. So to test this hypothesis, we decided in collaboration with Jean-Pierre Routy to do a Metformin clinical trial. What is Metformin? It's a drug that is used for diabetes, now is repurposed a little bit for cancer, and we were thinking repurposing it for HAV to block residual transcription. So indirectly, metformin can block mTOR, the mTOR pathway, and these are the people involved. So this is a collaboration with Jean-Pierre Routy and Nicolas Chamon. I'm not going to go into the results of this. This will be presented by my student, Augustin Firth, who is continuing the work that Delphine Plana is starting in my lab. Delphine is now working with Olivier Schwartz at Institut Pasteur in Paris. So please listen to her presentation that will be also virtual. So now talking again about retinoic acid, and since we were in the situation to test different drugs for their ability to block HAV reactivation, we were in the situation to modify to adapt a new viral outgrowth assay to simplify it so that we can do drug screening. So this is the reason we decided to do this work and try to make it more simple. So this is a paper that was just accepted in Frontiers in Microbiology after a long review process. We found that it is easier to recover viral replication competent virions if we split the cells every 3 days. And so from one well, we arrived with 8 wells after 12 days of activation in vitro. So, and also we demonstrated the importance of washing at day 3 to remove cellular factors that maybe have antiviral activity. So, so this is the performance of this assay. We're able to show that if we add retinoic acid, we can increase even further the viral outgrowth. And we were able, in collaboration with Nicolas Chamon and Marta Massanella, to transform this VOA into a QVOA, and this is the schematic representation of the protocol, and you can see that we can identify more infection units per million in the presence of ATRA. So then I will just make a kind of publicity to the work Amélie Catin is doing in the lab, who is finishing her PhD. She will also present this work as a poster. Mainly here, I would like to emphasize that we were able to identify CD16 positive monocyte-derived dendritic cells as cells expressing RALDH. This is the enzyme that metabolizes vitamin A in retinoic acid, and this activity is induced by triggers of toll-like receptor 2, such as Staphylococcus aureus. So I would like to make with this work that is in progress in the context of the CAHR collaboration, we would like to make a link between viral reservoirs in T cells and components of the microbiota that may be able to activate these reservoirs, and we would like to characterize the antigenic specificity of T cells carrying HAV reservoirs. So now this is the last part that will allow me to tell you a little bit more about the circadian clock machinery. So it started early in 2013 and 15 when we published some transcriptional characterization of Th17 and Th1, Th17 cells. And we were able to compare this transcriptome of these cells to the transcriptome of Th1 cells, and we were able to identify multiple differences and also similarities. But among differences, I would like to emphasize the differential expression of some transcription factors, which are PPAR gamma, ROR-C, ROR gamma T, and BMAL1 or aryl hydrocarbon receptor nuclear translocator-like. So let's discuss a little bit about PPAR gamma. PPAR gamma is very known as a transcriptional repressor, but also activator of multiple genes, is expressed in multiple cells, multiple tissues, is a therapeutic target for diabetes, for cardiovascular disease. And what was interesting for us was the fact that PPAR gamma is a repressor of ROR-C2 or ROR gamma T expression. In this way, it can have an impact on Th17 differentiation. So in 2013, we published this paper showing that PPAR gamma is an intrinsic negative regulator of HAV replication in Th17 cells. We showed that there is higher expression of PPAR gamma in Th1, Th17 cells. If when we did siRNA against PPAR gamma in primary CD40 cells, we could increase a viral integration in these cells, so confirming its role as a negative regulator. So then we looked at the nuclear localization of PPAR gamma after exposure to an agonist, which is rosiglitazone. And you can see there is a nuclear translocation of PPAR gamma, which is expected. And when we do this, we treat cells with rosiglitazone, there is an inhibition of viral replication in vitro. Then we also did the control experiment where we treated cells with rosiglitazone, which is an agonist, but also T0070907, which is an antagonist. And you can see that the nuclear translocation in this context doesn't happen anymore. So this opened our eyes toward a new idea, and we hypothesized that PPAR gamma antagonism promotes HAV reservoir activation and represents a new class of latency reversing agents for shock and kill strategies. So it was for us reasonable to assume that if PPAR gamma represses HAV, and it's known, it's well documented, and ROR-C, this is also well documented, if we block PPAR gamma activation with an antagonist, we will de-repress transcription. So we will increase transcription of both the virus and ROR-C dependent genes. So in a paper that was just accepted in Pathogens and Immunity, Delphine, together with other people in the lab, Augustine, and the idea was initially developed by Yue Zhang, who left the lab in 2017. So we're able to publish this paper and showing that indeed T007 increases IL-17 transcription, increases levels of cell-associated HAV RNA, but unexpectedly we observed a decreased release of virions from these cells. So we were a little bit confused. We went around right away to test this drug T007 in a viral outgrowth assay, and here we used the quadruplicates from the beginning so that we bypass we overcome the risk of the stochastic distribution of HAV reservoirs. So as you can see, exposure to T007 strongly reduces viral outgrowth without altering viability, and this is flow cytometry, while here we measure the HAV-P24 levels by ELISA, and we see a strong effect, inhibitory effect on viral outgrowth. So then we wanted to know if this effect can be seen also in CCR6 positive cells, and after infection. Indeed, we can see a significant decrease in HAV integration in CCR6 positive cells. This coincided with the upregulation of IL-17 expression. And we decided to do RNA-seq because we were a little bit lost in understanding what was going on with this drug. So we could see genes differentially expressed in the two situations. We could, by doing gene ontology and an interrogation of the NCBI interactome database, we could see that indeed the T007 upregulated genes involved in HAV transcription. CDK9 is one of them. So this explaining, therefore, an increased HAV transcription. But also we could observe the overexpression of multiple genes involved in antiviral responses. One example is IL-21, and we validated this at the protein level. IL-21 was interesting to us because we know there is paucity of IL-21 in infected people, and we know that in monkeys, if we supply with IL-21, as it was done by studies by Mirko Paiardini, we can restore mucosal immunity and decrease viral reservoirs. We know that IL-21 has antiviral effects through specific microRNA 29. So it made sense that T007 would upregulate IL-21 production. Another molecule that was upregulated was BST2. So BST2, as you all know, is involved in preventing the release of virions from infected cells. So with Augustine, we were able to show that cells identified HAV positive by HAV flow by using two different antibodies, express higher levels of P24 intracellularly based here on the MFI of P24 expression. But also infected cells will express significantly higher levels of BST2. And this was the case also for the uninfected bystander cells. So we observed an increase in BST2 expression. So this was for us mechanistic explanation of our observation. So we hope we can go further to test this PPAR gamma antagonist as a new strategy for HAV remission and or cure. Of course, in addition to multiple in vitro studies, we plan to do experiments in an in vivo model in humanized mice in collaboration with Ricoen, who is who has this platform and has a strong expertise in this field. So let me tell you about the aryl hydrocarbon receptor. You may think this is getting more and more complex. I will give you a very simple explanation why we got interested in aryl hydrocarbon receptor. This is another nuclear receptor, and it will regulate the expression of different genes. And it is capable to modulate the functions of different immune cells, such as dendritic cells and different subsets of T cells, including Th17 cells. So this aryl hydrocarbon receptor will bind to ligands that come from the external sources, including the food. So, and this is the reason there was this review article, You Are What You Eat, by Laura Hooper. And in fact, the main reason we decided to focus on AHR was its identification in mice and humans as a marker of non-pathogenic Th17 cells. We were for a long time thinking the existence or non-existence of non-pathogenic Th17 cells in HAV infection. And this was inspired a little bit by a publication by Branchley and Dweck showing in 2008 in a blood paper that in the lungs, Th17 cells are not depleted. So we hypothesized that maybe cells that are in the lungs are not permissive to infection. So we were still in search of non-pathogenic Th17 cells, and we're thinking that AHR may be a molecular mechanism underlying this resistance. It's interesting to highlight this paper by the group of Richard Flavelle, showing that AHR governs the Th17 transdifferentiation from inflammatory cells into IL-10 producing regulatory T cells. And for us, this was another reason to go into this hypothesis. we used a protocol to downregulate AHR, a CRISPR-Cas9 protocol that was published by other groups in 2016. And Debashri in the lab was able to make this work on primary cells. So we were able to knock down AHR expression in primary cells. We show decreased IL-22, IL-17, IL-10, and also integrating beta-7 expression without altering the expression of interferon gamma. This was for us highly interesting. We also could get similar results by using agonists and antagonists. And I would like to highlight the effect of the antagonist, the AHR antagonist CAHR2020, on the production of IL-22, IL-17, and IL-10 in primary CD4 T cells. Now we infected the cells with HIV and were able to see that the AHR knockdown silencing resulted in increased HIV replication. And this was observed by measuring integrated HIV DNA and also replication at HAVP24 level. We did in parallel experiments with the drugs, the agonist fix and the antagonist CAH22. And we could see that the antagonist would upregulate HIV replication in one representative donor, and this is in multiple donors. And we also compared this effect in CCR6 positive and negative T cells, and you can see that the replication is higher without any drug in the CCR6 positive, but CH22 upregulates HIV replication in CCR6 positive T cells. So this effect was not explained by an effect on CD4, CCR5, or CXCR4. So it was not linked to HIV entry. But at the post-entry level, when we used the VSVG pseudotyped HIV, we were also able to see differences, especially differences in HIV integration that was significantly increased in the presence of an AHR antagonist CH22. So the final experiment was to look at viral outgrowth from cells of R-treated people living with HIV. And you can see clearly that the antagonist will increase significantly the reactivation of HIV reservoirs by FACS, by flow cytometry, and also by ELISA. So what is AHR? So it was interesting that AHR was described to be a restriction factor for HIV in macrophages. This is work from the group of Paul Binash showing that AHR acts as a restriction factor in macrophages, but not in T cells. Why? Our explanation for this is that they used PHA IL-2 stimulation in CD4 T cells that does not induce AHR expression. And if you can see in this Western blotting, AHR expression is very low ex vivo, and it is induced by TCR triggering. So all our experiments were done with TCR triggering using CD3, CD28 antibodies. Another receptor we wanted to study is ROR-C, and this is the master regulator of Th17 cells. We benefited from a funding from CAHR, but most recently funding we received from the NeoMed in association with GSK. And we were able to demonstrate that indeed by using in this slide SCRNA in total CD4 T cells or in sorted CCR6 positive and negative, we can decrease HIV DNA, HIV replication and integration in cells after silencing ROR-C. Another way to approach this question was to look at viral replication in vitro and in cells that express or not ROR gamma T. And as you can see, the majority of HIV positive cells are located in the ROR-C2 positive fraction. We were also able to sort cells from R-treated individuals based on CCR6 and ROR-C2 expression and quantify HIV DNA. And as you can see, double positive cells are enriched in HIV DNA. And again, our final results we have here is that by using an antagonist for ROR-C, the GSK805, which is commercially available, we can decrease viral outgrowth from primary CD4 T cells. suggesting that this is a positive regulator of HIV replication. So I will finalize my presentation in the next 5 to 10 minutes by focusing on circadian variation in HIV reactivation during ART. I mentioned already other transcription factors. Now I would like to focus on more specifically on the circadian clock machinery. And as you can see, the circadian clock transcription factor, BMAL1 or RNTL, is overexpressed in Th17 versus Th1 cells. What is the circadian clock machinery? We have in the body a central clock and a peripheral clock. The central clock is influenced by light, food, and sleep. All the cells of the body express circadian clock machinery, mainly constituted from BMAL1 and clock. And this will bind to the E boxes in the promoters of different genes. And through a mechanism of positive and negative feedback, we will have circadian regulation of the expression of multiple genes. We know that circadian alterations are linked to exacerbated inflammation. We know that HIV infection is associated with circadian rhythm alterations. BMAL1 is a nuclear receptor. The studies published by Laura Hooper et al. Proposed a model in which BMAL1 will upregulate RevERB expression, which will repress NFIL3, which will repress RORγt. And in order to progress into this project, we started with a collaboration with the group of Nicolas Sermakian, who had access to BMAL1 knockout, and we could demonstrate that indeed, according to this model, BMAL1 knockout mice exhibit decreased IL-17 responses. By scRNA, we were able to show that IRNTL is a positive regulator of HIV DNA integration in primary T cells. By overexpressing BMAL1 using viral antivirus, we were able to show the same, an increased viral replication in primary CD4 T cells. We consider this model may be valid. Studies published by the group of Sharon Lewin provide more evidence for the direct implication of BMAL1 in the direct regulation of HIV transcription. So another component of the circadian clock machinery is, as I said, it's RevERB alpha. And Christ, who is a PhD student in the lab, could demonstrate that in contrast to BMAL1 that is typically increased with with TCR triggering, and this is consistent with the fact that cells need TCR triggering to get infected. In contrast, RevERB alpha and beta, they are decreased with TCR triggering, and this is consistent with the idea that these are transcriptional repressors. So in collaboration with Laura Hooper, we had access to some drugs that are agonists for RevERB alpha and beta, SR9009 and 9011. And Christ was able to demonstrate that by treating cells with this RevERB agonist, we could decrease IL-17 production and decrease interferon production. This was a little bit surprising because based on the model of Laura Hooper, we expected the opposite. He also could see in a viral outgrowth assay that there is a decrease reactivation of HIV reservoirs or viral outgrowth in the presence of this RevERB agonist. So suggesting that RevERB is a negative regulator of HIV transcription, directly or indirectly. So this didn't fit too much with the model proposed by Laura Hooper, but fitted much better with the most recent model proposed by Laura Salt, a collaborator of us, where she's showing that RevERB alpha acts as a repressor of the transcription of multiple genes that express ROR responsive elements in their promoters. So of course, we would like to test all these drugs in humanized mice. I'd like to point to the fact that we started this collaboration with Cholo Chong. It's been 4 years today. He left us. he was helping us to show that indeed when we infect the mice, viral replication goes up and Th17 cells are depleted. So showing that the humanized mice model is a good model to study Th17 responses in vivo. So now to finish with the last experiment, which is done with the people living with HIV. It's for me, I'm limited in time. I cannot explain to you what a adventure was the organizing of this small clinical trial, if I can call it like this, where we hospitalized 11 infected people, and we draw blood every 4 hours for 24 hours to collect 7 samples per patient for plasma soluble factor quantification and also virological immunological measurements in PBMCs. This is the cohort we used. They were all men, 11. It was a fantastic experience for me to discuss with them and to understand better their medical conditions and to understand that in fact every donor was a different one, a different case. So we could show in these patients that there is typical cortisol variation in the plasma. There is also typical variation of melatonin in the plasma during the day. We also could observe for the first time that there were daily variations in FABP and LPS binding proteins in the plasma. These are 2 markers associated with the leaky gut. And we were able to do FACS analysis on the fresh blood and to look at the frequency of different cells. And we confirmed the similar to what was published in HIV negative donors. There is a rhythmicity in the peripheral blood in terms of counts of different subsets, CD3 positive, CD4, CD8, and also regulatory T cells. I'm not going to go into details. I will tell you that we still need to finish the study. What we want to do is to look at residual viral replication during the day by different virological methods and to try to find molecular mechanism associated with these changes, if there are any changes, using RNA-seq and also validations at the RNA and protein levels. So as I said, every patient was a distinct one. We try to understand this heterogeneity and to take it into account. So now to conclude, this is one figure I submitted when I first submitted this grant in 2016 and it was rejected. So because it was too complex, but unfortunately we are in this situation of complexity. I was mentioning multiple transcription factors that are circadian components, BMAL1, others that are circadian regulators, RevRB, RORC2, PPAR gamma, AHR. And then I also talked to you about mTOR, which is a molecule that is regulated in its expression in a circadian way. So of course, I can talk more and more about this. I will finish with a conclusion that early diagnosis, early art, and a healthy lifestyle, together with alternative strategies specifically designed to protect Th17 cells from depletion and functional alterations, will contribute to HIV eradication. We identify some new potential drug targets, and I hope we will have the strength and the courage to go further with this testing. And I would like to also emphasize that prevention is important to stop these epidemics. So here I'm gonna really finish by thanking everybody that participated to this work. As you can see, I didn't do this alone. I had the help of many students in my lab, and especially the help of my collaborators, and especially the help of my clinicians. And I would like to emphasize the wonderful collaboration established within CAHR that allowed me to grow. And of course, the money we receive from different organizations, including the CAHR and FRQS and NIH, and most recently NeoMed and GSK. Thank you very much for your attention.